we now turn our attention to a group of protists that in general we call them Amoeboid Protists. However, this is not a monophyletic group. Protists that move using pseudopodia or false feet, which will be the literal translation for this type of locomotion, are going to be classified into two different groups of protists. The first one, and you can see those on the screen here, are going to be the Amoeboid Protists of the subgroup Ryzeria. This is a subgroup of SAR, S-A-R. The R in SAR is going to be for Ryzeria. And if you think about the word, Ryze means the same as root, which would be kind of like a long and slender type of a structure. Now, in the case of the Ryzerians, as protists, these extensions that are thin and slender are not going to be for anchoring. These are actually for locomotion. So these are the pseudopodia. With the help of a light microscope, these actually look like thin crystalline needles. But they can be extended or retracted, and as I said just a moment ago, they're going to be aiding for locomotion. And so among the Ryzerians, we're going to see the Radial Ariens, the Foraminiferans, and the Cercozoans. Let's focus for a moment on the Radial Ariens and the Foraminiferans. Oh, by the way, I wanted to use this illustration because it's enlarged enough that we can see one of these examples of Ryzerians. This here is a type of a foraminiferan. What we see here is actually kind of like a protective structure for the cell. And this protective structure is made of calcium carbonate, a type of mineral that is abundant in seawater. And the little tiny holes you see here are going to be the perforations, will be the little tiny openings through this protection for the cell that the rhizopods, that these pseudopods are going to be moving out. So when you look at this illustration and you see all the pseudopodia like thin, slender rays, they're actually projecting out of these holes on the foraminiferan test, which is, like I said, this protective structure for the foram cell. Let's go ahead and take a look at the Radial Ariens. These are by far my favorite single cell protists because of the intricate and beautiful details in the tests that they produce. I'm going to do this enlargement again so you can see one of those Radial Ariens, and you can see the ray-like, thin, slender, long pseudopodia. Remember, these are structures that are used for locomotion, but they can also be used for the capturing of prey, other single-cell plankton that is abundant in the ocean, and these kinds of protists will be depending on as a source of energy and nutrition. Now you can see here the remnants of these Radial Ariens. We only have here the tests. In the case of Radial Ariens, the test is actually made of silica. It's a carbon type of compound, but it doesn't have the calcium component of it. So silica is real similar to the same material that makes glass. And look at the patterns, look at the shapes. I mean, some of these things look like some form of an alien ship. Others are just going to maybe remind you of like a disco ball, but they're all pretty beautiful, impressive, the kinds of details and patterns you can observe in the tests of Radial Aria. They're going to be, these pseudopodia will be emerging through the body, and as I said, pseudopodia will be useful in both locomotion and also for the capturing of food. Now we can see the foraminiferans. These are the ones where the tests are made of calcium carbonate. And they also make beautiful patterns, shapes. And whenever you see the little tiny pores, remember that those pores are going to be the openings for the pseudopodia to move out and extend. Some of these foraminiferans are going to hold within also endosymbiotic algae that can help them with obtaining sugar by the process of photosynthesis. And so the foram provides a safe space for algae cells to live in, and then in return, they get some of the sugar made by the photosynthetic activity of algae. Okay, so these are going to be the amoeboid protists of the clade Rhizaria, which is a subgroup of the supergroup ZAR. Now let's move, shift our attention to the uniconta. These are going to be amoeboid protists that are in the same group as other organisms that you may not anticipate to find here. So the uniconta, it's a clade that includes also animals and fungi. We will be turning our attention to fungi after we complete our study of the protists. But it's interesting that not only do we have here protist kinds of organisms among the uniconts, but we're also going to see other complex multicellular organisms such as the animals. The root for the uniconts is a controversial one, is one that scientists are not quite certain what would be the common ancestor to all of these groups that share, among many things, similarities at the molecular level, such as similarities in protein and DNA and messenger RNA structure. One of those types of amoeboid protists that are among the uniconts would be the slime molds. These slime molds are also known as the acellular molds, or acellular slime mold. Let me be more specific. We call them acellular because when you find these in a forest, and by the way, if you're curious and you take a hike through a forest, if there's an abundance of leaf litter, I dare you to lift up some of those leaves and you may find slime molds like the one you see right here. We call these acellular slime molds because it's in reality many cells that have been made by the process of mitosis. However, there is no plasma membrane dividing the cells. So a close inspection will reveal it's like a huge mass of cytoplasm, and there's going to be many diploid nuclei in every one of these mesh-like network types of acellular slime mold. What happens sometimes is that the conditions turn not as favorable as the slime mold itself would hope to have, and that when conditions begin to turn harsh for them, some of the cytoplasmic masses and nuclei begins to form kind of like a bell, kind of like a structure, kind of like a vesicle, and in here, a few of those nuclei are going to turn into what we call a sporangium. Remember, angium, I've described this to you before, means vessel, and spore, well, it's a reproductive structure. So some of those nuclei are going to make a capsule, and they are going to become spores. The spores here are going to undergo meiosis, and before they are, released, the spores are going to be haploid, and when the spores are going to be germinating, they're going to be emerging into haploid amoeba cells. And so these amoeboid cells then go through the process of fertilization. There are two kinds of amoeboid cells. Maybe it will be easier for you watching this video if I can enlarge this. There are two kinds of cells that can emerge from a spore. Some can be amoeboid, some can be flagellated. And so they usually pair up. Two flagellated cells are what is needed to undergo fertilization, or two amoeboid cells are also joining each other. That's fertilization. That makes a diploid zygote, and this begins to go through cell division again. The nucleus multiplies, but there's no plasma membrane dividing the two nuclei. And so this turns into the feeding plasmodium, where mitosis continues, but only dividing the nucleus. However, the cytoplasm never gets to be divided, and it goes in search of food, decomposing organic matter found at the forest floor. That's going to be the cell cycle for the acellular slime molds. Remember, we call them acellular because when you look at one of these masses of a feeding plasmodium, it's just going to be many diploid nuclei, but there's no plasma membrane dividing those nuclei. We also have cellular slime molds, and the cellular slime molds are going to be forming multicellular aggregates. Let's begin exploring their life cycle here with one of these aggregations of amoeba types of organisms. And so they are going to be migrating and feeding and migrating and feeding. And then at some point, they are going to be forming a fruiting body. Inside of the fruiting body, some of these cells will harden on the outside, forming like a capsule that is going to be responsible for holding, producing, and eventually releasing spores. The spores are haploid. As you know, there's only one set of chromosomes. And when the little amoebas emerge, they are going to live solitary. Now, these solitary amoebas also go through the process of feeding. Two amoebas may fuse and engage in sexual reproduction. And so when that happens, the two amoeba fuse, they become a zygote. Then this outer covering hardens. Inside, the nucleus is going to undergo meiosis, forming four nuclei, and those nuclei eventually continue to dividing furthermore, forming more of these amoebas. The solitary amoebas can engage again in sexual reproduction, or they can make one of these aggregates that will continue to migrate until they go through the process of asexual reproduction where spores are going to be made. And so the reason we call these cellular slime molds is because all of these amoebas in this aggregate each are made up of a nucleus, and they have their own plasma membrane surrounding each of the amoeba cells. One of the examples that is commonly mentioned in a class like this will be Dictyostelium, and like I said, this is part of this supergroup Uniconta. The Unicons also are going to include the gymnamoebas. The name has been recently changed to the tubulinids. And so these are going to be amoeba-like organisms, like the ones we study in a lab, where the pseudopodia are going to be broad, sometimes more like a lobe-like type of a pseudopodium, and they move using a cytoplasmic streaming, where masses of cytoplasm move in one direction, aided by the contraction of protein fibers such as microfilaments of actin. Actin will be the same type of a protein we find in our muscle cells and used for locomotion. These tubulinid amoebas are going to be heterotrophic. They like to feed on other protists, like Paramecia. You can find them feeding on algae. You can find them feeding also on bacteria. So this is not a type that is going to be known to be a dangerous type of a parasite. You might have heard that amoebas are parasites of the gastrointestinal system of many mammals, including humans. Others can be severe parasites that can move into the nervous system when they can be lethal as well. That is usually going to be the case of the entamoebas. These entamoebas are not going to have the same type of long pseudopodia. They do have an amoeboid type of movement, but not the long extensions of the cytoplasm. Here you see a picture of Entamoeba histolica. This is one that is notorious for causing a gastrointestinal disease, which can be severe, known as amoebic dysentery. And it's going to be the third leading cause of deaths in humans around the world, amoebic dysentery. So entamoebas, we also have the gymnamoebas, or tubulinids as they are now called, and then we also have cellular slime molds and the acellular slime molds, all being part of this supergroup we call the Unicons. That is all with regards to amoeboid protists that I wanted to share with you.